Evolution of prokaryotes, the origin and diversification of Earth's first life forms. The evolution of prokaryotes represents the earliest chapter in the history of life on Earth, beginning around 3.8 to 4 billion years ago. Prokaryotes were the first cellular life forms, arising from self-replicating molecular systems encapsulated in lipid membranes. These primitive life forms lacked a nucleus and membrane-bound organelles, characteristics that still define modern bacteria and archaea. The evolution is critical for understanding the origin of life, early metabolic pathways, and the biochemical foundation upon which all other life evolved. The earliest prokaryotes likely originated from protocells, self-assembled lipid vesicles encapsulating catalytic RNA and other molecules. With the advent of genomic stability through DNA, coupled with RNA-based catalysis and rudimentary ribosomes, the first true prokaryotic cells were formed. These organisms were most likely anaerobic heterotrophs, relying on environmental organic molecules for energy and carbon, given the absence of atmospheric oxygen in early Earth conditions. The metabolic capabilities of early prokaryotes were shaped by natural selection acting on molecular variation, leading to innovations such as fermentation, anaerobic respiration, and later, chemolithotrophy, the ability to extract energy from inorganic molecules like hydrogen sulfide or iron compounds. The evolution of autotrophic prokaryotes marked a significant leap. Oxygenic photosynthetic bacteria, such as green sulfur bacteria, which use light energy but not water as an electron donor. Eventually, the appearance of oxygenic photosynthesis, pioneered by cyanobacteria, fundamentally transformed the biosphere. Cyanobacteria use water as an electron donor, releasing oxygen as a byproduct, a process that culminated in the Great Oxygenation Event, around 2.4 billion years ago. This event not only increased atmospheric oxygen but also created selective pressure for organisms to evolve oxygen-tolerant and later aerobic metabolic pathways, such as oxidative phosphorylation. It also led to the formation of the ozone layer, shielding Earth from UV radiation and permitting more complex life forms to evolve. Prokaryotic evolution also includes the divergence of the two major prokaryotic domains, bacteria and archaea, which, despite sharing a prokaryotic cell structure, differ in membrane lipid composition, ribosomal structure, and gene expression mechanisms. Archaea often inhabit extreme environments, extremophiles, and share several molecular and genetic features with eukaryotes, suggesting an important evolutionary link between archaea lineages and the origin of eukaryotes. Phylogenetic studies based on ribosomal RNA sequencing by Carl Woese revealed that life is divided into three domains, bacteria, archaea, and eukarya, with archaea and eukarya forming a closer evolutionary group than either with bacteria. Prokaryotes are also characterized by horizontal gene transfer (HGT) mechanisms, such as transformation, transduction, and conjugation, which have profoundly influenced their evolutionary trajectory. HGT allows rapid acquisition of new traits, such as antibiotic resistance or metabolic innovations, enabling prokaryotes to adapt swiftly to environmental changes. This complicates traditional tree-like models of evolution, suggesting instead a network-based or web-like evolution in early life. Prokaryotic evolution is also supported by microfossil evidence in stromatolites, layered structures formed by microbial mats, which date back over 3.5 billion years. These fossil records confirm the long-standing dominance of prokaryotic life on Earth. Despite their microscopic size, prokaryotes played and continue to play a central role in biogeochemical cycles, nutrient recycling, and climate regulation, shaping the environment in which all other life evolved. In the CSIR-NET Life Sciences exam, questions from this topic frequently test the timeline of events, metabolic evolution, photosynthesis origins, phylogenetic relationships, and significance of HGT. It is also linked with topics like origin of eukaryotes, molecular phylogeny, microbial ecology, and bioenergetics. Previous year CSIR-NET questions. CSIR-NET December 2013. Which of the following is considered the most ancient metabolic process in prokaryotic evolution? A. Oxygenic photosynthesis. B. Aerobic respiration. C. Fermentation. D. Chemosynthesis using methane. Answer. C. Fermentation. CSIR-NET June 2017. Cyanobacteria are significant in the evolutionary history of life because they are the ancestors of modern viruses. B. Were the first to use organic molecules as electron donors. C. Introduced oxygen into Earth's atmosphere. D. Are eukaryotes with prokaryotic traits. Answer. C. Introduced oxygen into Earth's atmosphere. CSIR-NET December 2020. Archaea differ from bacteria in all of the following aspects except a. Membrane lipid composition. B. Presence of peptidoglycan. C. Sensitivity to antibiotics. D. Ribosomal RNA sequence similarity to eukaryotes. Answer. B. Presence of peptidoglycan. Would you like to continue with the next lecture, Evolution of Eukaryotes, Endosymbiotic Theory, or RNA World and Ribozymes? These topics are directly connected and frequently asked in CSIR-NET Life Sciences. You said. Origin of eukaryotic cells, ChatGPT said. Here is your CSIR-NET JRF level, highly detailed, maximum word limit lecture on Origin of eukaryotic cells, crafted as per your exact requirements. No tables. Paragraph format only. Full coverage from base to advanced level. Previous year CSIR-NET questions with answers in years. Acts as your complete and only coaching material for this topic. Origin of eukaryotic cells, a major evolutionary transition. The origin of eukaryotic cells marks one of the most profound transitions in the history of life, enabling the rise of complex multicellular organisms. Eukaryotic cells are characterized by the presence of a membrane-bound nucleus, organelles such as mitochondria and plastids, linear chromosomes, and an advanced cytoskeleton. Origin of eukaryotic cells, a major evolutionary transition. The origin of eukaryotic cells marks one of the most profound transitions in the history of life, enabling the rise of complex multicellular organisms. Eukaryotic cells are characterized by the presence of a membrane-bound nucleus, organelles such as mitochondria and plastids, linear chromosomes, and an advanced cytoskeletal network. These features contrast sharply with prokaryotic cells, and the emergence of such complexity required a series of evolutionary innovations. The widely accepted explanation for the origin of eukaryotic cells is the endosymbiotic theory, first proposed by Lynn Margulis, which suggests that eukaryotes evolved through a symbiotic relationship between ancestral prokaryotic cells. Molecular and fossil evidence suggests that the first eukaryotic cells emerged approximately 1.8 to 2 billion years ago. This event followed the appearance of oxygenic photosynthesis in the Great Oxygenation Event, which increased atmospheric oxygen and enabled the evolution of aerobic respiration. The endosymbiotic theory proposes that key organelles in eukaryotes, especially mitochondria and plastids, originated as free-living bacteria that were engulfed by a larger host prokaryote, possibly an archaeal cell. Over time, these engulfed cells established a mutualistic relationship, transferring some of their genes to the host genome and becoming permanent organelles. Mitochondria are believed to have originated from an ancestral alpha proteobacterium, a type of aerobic bacterium capable of efficient energy production through oxidative phosphorylation. The host cell, likely an archaeal organism with a rudimentary cytoskeleton, engulfed the bacterium via phagocytosis or a related mechanism. Instead of digesting it, the host maintained the endosymbiont, which provided ATP in return for protection and nutrients. This led to the mitochondrion's integration, gene transfer to the host's nuclear genome, and the loss of many independent features of the engulfed bacterium. Evidence supporting this theory includes the presence of circular DNA, double membranes, 70S ribosomes, and binary fission in mitochondria, all reminiscent of bacterial ancestry. The origin of plastids, e.g., chloroplasts in photosynthetic eukaryotes, is attributed to a secondary endosymbiotic event, where a eukaryotic host cell engulfed the cyanobacterium, the ancestor of modern chloroplasts. Like mitochondria, chloroplasts possess their own circular DNA and prokaryotic-like ribosomes and reproduce by binary fission. In some such as dinoflagellates and euglenids, tertiary endosymbiosis occurred, involving the engulfment of eukaryotic algae by other eukaryotes, adding additional layers of complexity. Besides organelle acquisition, the evolution of eukaryotes involved the development of a complex cytoskeleton, enabling dynamic shape changes, intracellular transport, and advanced meiotic division. The evolution of endomembrane systems like the nuclear envelope, Golgi apparatus, and endoplasmic reticulum may have resulted from invaginations of the plasma membrane, leading to compartmentalization and greater regulatory control over cellular processes. The emergence of meiosis and sexual reproduction added genetic diversity and accelerated evolutionary innovation in eukaryotes. Recent advances in genomics have revealed that Asgard-Archaea, a newly discovered group of archaea, share numerous eukaryotic-like genes and are considered the closest extant relatives of eukaryotes. This discovery strengthens the hypothesis that eukaryotes evolved from a syntrophic relationship between an archaeal host, likely Asgard-related, and a bacterial endosymbiont mitochondrial ancestor. The fusion of archaeal informational processing systems replication, transcription with bacterial metabolic genes led to the chimeric nature of eukaryotic genomes. From the CSIRNet perspective, this topic links multiple core areas: evolutionary biology, molecular biology, genetics, and cell biology. Questions often test understanding of endosymbiotic theory, features of mitochondria and plastids, the timeline of events, genetic evidence, and distinctions between prokaryotic and eukaryotic features. One must also be aware of secondary and tertiary endosymbiosis, the genomic complexity of eukaryotes, and the molecular signature of endosymbiotic origins. Previous year CSIRNet questions. CSIRNet June 2015. Mitochondria are believed to have originated from a gram-positive bacteria B, cyanobacteria C, alpha proteobacteria D, actinobacteria. Answer: C, alpha proteobacteria. CSIRNet December 2014. Which of the following is not a piece of evidence supporting the endosymbiotic origin of mitochondria and chloroplasts? A, presence of circular DNA. B, ADS ribosomes. C, double membranes. D, reproduction by binary fission. Answer: B, ADS ribosomes. CSIRNet June 2018. Which of the following supports the idea that eukaryotic genomes are chimeric in nature? A, eukaryotes possess only archaeal genes. B, eukaryotes possess only bacterial genes. C, eukaryotic informational genes are archaeal, and metabolic genes are bacterial. D, eukaryotic ribosomes resemble those of eukaryotic mitochondria. Answer: C, eukaryotic informational genes are archaeal, and metabolic genes are bacterial. Shall we proceed with the next lecture? Endosymbiotic theory in detail, evidences for evolution, or RNA world and the transition to DNA protein world. Let me know what topic you want next, I'll deliver it with the same quality. You said. Evolution of unicellular eukaryotes. ChatGPT said. Here is your CSIRNet JRF level, ultra detailed, paragraph format lecture on evolution of unicellular eukaryotes, tailored as your exclusive coaching material, adhering strictly to your format. No tables. Fully written in paragraphs, using maximum word limit. Covers all key areas from basics to advanced level. Based on important concepts tested in CSIRNet. Includes 2 to 3 previous year CSIRNet questions with answers in years. Evolution of unicellular eukaryotes, a foundation of eukaryotic diversity. The evolution of unicellular eukaryotes marks a pivotal moment in the history of life, bridging the evolutionary gap between simple prokaryotic life forms and the complex multicellular organisms that dominate today's biosphere. These unicellular eukaryotes, traditionally referred to as protists, exhibit remarkable diversity in morphology, metabolism, locomotion, and reproduction. They were the first organisms to combine the internal complexity of eukaryotic cells, such as membrane-bound organelles, a true nucleus, and a cytoskeleton, with the simplicity of unicellular existence, enabling novel adaptive strategies that were impossible for prokaryotes. The earliest unicellular eukaryotes appeared approximately 1.8 to 2 billion years ago, during the Proterozoic eon, following the origin of mitochondria via endosymbiosis and the subsequent development of complex intracellular organization. The key event that laid the foundation for the evolution of unicellular eukaryotes was primary endosymbiosis, whereby an ancestral archaeal host engulfed an alpha proteobacterium that became the mitochondrion. This host endosymbiont integration enabled oxidative phosphorylation, leading to higher energy efficiency. The evolution of a dynamic cytoskeleton, intracellular trafficking systems, linear chromosomes with histones, and compartmentalization of genetic material into a nucleus allowed for greater genomic and regulatory complexity. These adaptations provided a selective advantage in terms of environmental flexibility, phagocytic feeding, and cellular control mechanisms. Unicellular eukaryotes diversified into numerous lineages, which are now classified into major eukaryotic supergroups: Excavata, Amoebozoa, Archaeoplastida, SAR, Straminopols, Alveolates, Rosaria, and Apis tocunda. This classification is based on phylogenetic data derived from molecular analyses, such as rRNA sequences and conserved gene families. For instance, excavates include flagellated protists like Giardia and Euglena, many of which possess modified mitochondria or rely on anaerobic metabolism. The SAR clade includes ciliates, diatoms, and foraminifera, with unique adaptations like alveoli under the plasma membrane or intricate silica-based skeletons. Archaeoplastida, which gave rise to plants, includes unicellular algae like Chlamydomonas. These organisms acquired plastids through a separate endosymbiotic event involving cyanobacteria, further increasing metabolic versatility. One of the hallmark features of many unicellular eukaryotes is their flexible modes of nutrition. They can be autotrophic, e.g., euglena, heterotrophic, e.g., amoeba, or mixotrophic, e.g., dinoflagellates. This metabolic diversity is possible because of complex intracellular compartments and vesicle-mediated transport systems. Locomotion is facilitated by structures such as flagella, cilia, or pseudopodia, powered by cytoskeletal components like microtubules and actin filaments. Additionally, some groups possess contractile vacuoles for osmoregulation, a function absent in prokaryotes. The reproductive strategies of unicellular eukaryotes include both asexual, binary fission, budding, and sexual reproduction, syngamy followed by meiosis, allowing for genetic recombination and rapid evolutionary adaptation. The evolution of meiosis and syngamy in unicellular forms is considered a key innovation that later enabled multicellularity. Moreover, many unicellular eukaryotes can form dormant cysts or spores, allowing them to survive unfavorable conditions, adding to their ecological resilience. From an evolutionary perspective, unicellular eukaryotes were the precursors to all eukaryotic multicellular life forms. Some lineages, such as Volvocene algae, demonstrate a clear evolutionary transition from unicellularity to multicellularity, making them model systems for studying the origins of complex body plans. Others, like choanoflagellates, are closely related to animals and help trace the origins of metazoans. Hence, studying unicellular eukaryotes is critical to understanding both early eukaryotic evolution and the genetic, molecular, and cellular innovations that led to multicellular complexity. Molecular clock data and fossil records, such as microfossils and biomarker lipids, support the timing and diversity of early eukaryotic evolution. The presence of steranes, sterile-derived molecules, in Precambrian rocks provides biochemical evidence of eukaryotic life dating back nearly 2.7 billion years, though morphological evidence is typically dated later. Genomic analyses of modern unicellular eukaryotes reveal horizontal gene transfer, gene duplication, and genome streamlining, all of which played key roles in shaping early eukaryotic evolution. In CSIRNet JRF Life Sciences, this topic is frequently tested through questions related to eukaryotic origins, protist diversity, organelle function, and phylogenetics. One must understand the distinctions between different eukaryotic lineages, the endosymbiotic origins of organelles, structural adaptations, and the role of unicellular organisms in evolutionary transitions. Previous year CSIRNet questions. CSIRNet June 2016. Which of the following is the closest unicellular relative of animals, providing clues to the origin of multicellularity? A. Chlamydomonas. B. Dictyostelium. C. Choanoflagellates. D. Trypanosoma. Answer: C. Choanoflagellates. CSIRNet December 2015. Many protists can reproduce both sexually and asexually. What is the main evolutionary advantage of sexual reproduction in unicellular eukaryotes? A. Faster population growth. B. Generation of genetic diversity. C. Conservation of energy. D. Prevention of extinction. Answer: B. Generation of genetic diversity. CSIRNet June 2020. Which of the following supports the endosymbiotic origin of plastids in unicellular algae? A. Plastids contain circular DNA and 70S ribosomes. B. Plastids lack membrane. C. Plastids contain linear DNA and 80S ribosomes. D. Plastids divide by mitosis. Answer: A. Plastids contain circular DNA and 70S ribosomes. Would you like the next lecture on transition to multicellularity, RNA world hypothesis, or any other specific subtopic? Let me know and I'll prepare it with the same thoroughness. You said. Anaerobic metabolism. ChatGPT said. Here is your CSIRNet JRF Life Sciences Ultra Detailed Lecture on Anaerobic Metabolism, written in maximum word limit, no tables, paragraph format only, and tailored to CSIRNet level with deep conceptual clarity, previous year question areas, and solved questions from the exam. Anaerobic metabolism, cellular energy generation in the absence of oxygen. Anaerobic metabolism is a central bioenergetic process in cells that enables the production of ATP in environments devoid of molecular oxygen. It is one of the most ancient and evolutionarily conserved metabolic strategies and preceded aerobic respiration in early Earth's anoxic atmosphere. Anaerobic metabolism allows organisms to survive and thrive in hypoxic or anoxic niches, such as deep sediments, gastrointestinal tracts, or decaying organic matter. It plays a vital role in both prokaryotic and eukaryotic cells under oxygen-limited conditions and involves different biochemical pathways such as fermentation and anaerobic respiration. Importantly, anaerobic metabolism is not a mere backup system for oxygen deprivation but a distinct and regulated form of energy production, central to the metabolism of numerous organisms across the domains of life. In glycolysis, glucose is catabolized to pyruvate through a series of enzyme-catalyzed steps in the cytoplasm. This initial stage of anaerobic metabolism yields 2 ATP molecules for glucose via substrate-level phosphorylation. In the presence of oxygen, pyruvate enters mitochondria for oxidative phosphorylation, but in anaerobic conditions, it is diverted to alternate fates that regenerate NAD from NADH, allowing glycolysis to continue. The fate of pyruvate varies depending on the organism and environmental conditions. In lactic acid fermentation, typical of muscle cells and certain bacteria, e.g., lactobacillus pyruvate is reduced to lactate by lactate dehydrogenase. In alcoholic fermentation, as seen in yeasts like Saccharomyces cerevisiae, pyruvate is first decarboxylated to acetaldehyde, which is then reduced to ethanol. Both processes regenerate NAD but yield no additional ATP beyond glycolysis. Anaerobic respiration, unlike fermentation, involves the use of electron transport chains, ETCs, but with terminal electron acceptors other than oxygen. This process is widely used by prokaryotes such as bacteria and archaea in oxygen-deprived environments. Instead of oxygen, organisms may use nitrate, sulfate, sulfate, fumarate, carbon dioxide, CO, or even metal ions as final electron acceptors. For example, Pseudomonas denitrificans uses nitrate reduction to produce nitrogen gas denitrification, while disulfovibrio uses sulfate to produce hydrogen sulfide. Anaerobic respiration yields more ATP than fermentation but is generally less efficient than aerobic respiration. These processes are tightly coupled to membrane-bound ATP synthase via proton gradients, illustrating the diversity and sophistication of life even in the absence of oxygen. From an evolutionary perspective, anaerobic metabolism likely dominated in early cellular life forms. Before the Great Oxygenation Event, approximately 2.5 billion years ago, Earth's atmosphere was largely reducing, and prokaryotes evolved complex biochemical pathways to exploit a wide array of redox reactions. The appearance of oxygen was toxic to many early anaerobes, leading to the evolution of antioxidant defenses or the occupation of anaerobic ecological niches. Anaerobic metabolism remains a core feature in modern biology, not only as a relic of evolutionary history but also as a functional necessity in various specialized cells and organisms. For instance, certain parasites like Giardia lamblia and Amoeba histolytica possess modified mitochondria known as mutasomes or hydrogenosomes, which facilitate anaerobic metabolism through substrate-level phosphorylation and hydrogen production. In metazoans, anaerobic metabolism becomes critical during high energy demands or hypoxic conditions. During intense muscular activity, oxygen delivery to muscles may become insufficient, triggering lactic acid fermentation. This temporary shift enables continued ATP production but leads to lactate accumulation, which must later be metabolized in the liver via the Cori cycle. In cancer biology, many tumor cells exhibit the Warburg effect, where cells preferentially use glycolysis and lactic acid fermentation even in the presence of oxygen, aerobic glycolysis, possibly to support rapid proliferation and adaptation to hypoxic tumor microenvironments. In ecological terms, anaerobic metabolism contributes to global biogeochemical cycles. For example, methanogenesis by archaea such as methanobacterium produces methane, a potent greenhouse gas, under strict anaerobic conditions. Sulfate-reducing bacteria contribute to sulfur cycling and can also lead to biocorrosion. Anaerobic decomposition in wetlands and animal beds plays a vital role in carbon recycling. These processes also find applications in biotechnology, including anaerobic digestion for biogas production and industrial fermentation for ethanol or lactic acid production. From a CSIR-NET Life Sciences examination standpoint, understanding anaerobic metabolism involves key concepts such as substrate-level phosphorylation, fermentation. From a CSIR-NET Life Sciences examination standpoint, understanding anaerobic metabolism involves key concepts such as substrate-level phosphorylation, fermentation pathways, redox balancing, energy yield comparisons, microbial diversity, and evolutionary relevance. Questions often focus on distinguishing between fermentation and anaerobic respiration, the molecular fate of pyruvate, alternative electron acceptors, and adaptations of anaerobes. CSIR-NET previous year questions. CSIR-NET June 2017. In anaerobic respiration, which of the following can serve as a terminal electron acceptor in certain bacteria? A. Glucose. B. Oxygen. C. Nitrate. D. ATP. Answer: C. Nitrate. CSIR-NET December 2016. During anaerobic fermentation of glucose in muscle cells, the immediate purpose of converting pyruvate to lactate is to a. Remove pyruvate from a cell. B. Produce more ATP. C. Generate COD. Regenerate NAD. Answer: D. Regenerate NAD. CSIR-NET June 2021. Which of the following statements correctly describes the difference between fermentation and anaerobic respiration? A. Only fermentation involves an electron transport chain. B. Fermentation uses oxygen as the terminal electron acceptor. C. Anaerobic respiration yields more ATP than fermentation. D. Fermentation yields more ATP than aerobic respiration. Answer: C. Anaerobic respiration yields more ATP than fermentation. Shall I proceed with the next lecture on ATP synthesis and energy yield, or would you like to cover oxidative phosphorylation, mitochondrial evolution, or any other topic from your CSIR-NET syllabus? Let me know, I'll make it just as exhaustive and exam-focused. You said. Photosynthesis and aerobic metabolism. ChatGPT said. Memory updated. Here is your CSIR-NET JRF. Here is your CSIR-NET JRF Life Sciences Detailed Theory lecture on photosynthesis and aerobic metabolism, written in an ultra-comprehensive, paragraph-based, and exam-oriented style, with maximum depth, note tables, and solved CSIR-NET previous year questions. This note is crafted to be your only source of preparation for this topic. Photosynthesis and aerobic metabolism. Photosynthesis and aerobic metabolism represent the two fundamental and opposing pillars of the biological energy economy. Together, they form a bioenergetic cycle that sustains almost all life on Earth, with photosynthesis capturing solar energy and converting it into chemical energy, and aerobic metabolism harnessing this stored energy to perform cellular work. Understanding these processes is critical not only for CSIR-NET but also for mastering core concepts in cell biology, biochemistry, and plant physiology. Photosynthesis, the light-dependent conversion of solar energy. Photosynthesis is the process by which green plants, algae, and cyanobacteria convert carbon dioxide and water into glucose and oxygen using sunlight. It occurs primarily in the chloroplasts and involves two major stages, the light reactions and the Calvin cycle, dark reactions. The light reactions occur in the thylakoid membranes and involve two photosystems, photosystem II PSII and photosystem I PSI. These photosystems contain chlorophyll and accessory pigments which absorb light, excite electrons, and drive their flow through the Z scheme, ultimately resulting in the generation of ATP and NADPH via photophosphorylation. In non-cyclic photophosphorylation, electrons travel from water through PSII and PSI to NADP, producing NADPH and releasing O from water. In cyclic photophosphorylation, electrons from PSI return to the plastoquinone pool and are cycled back, producing ATP but not NADPH or O. This cyclic pathway allows the plant to adjust the ATP-NADPH ratio depending on metabolic needs. The Calvin cycle, occurring in the stroma, uses ATP and NADPH to fix CO into 3-phosphoglycerate via rubisco, eventually synthesizing triasphosphates that are precursors to glucose. This cycle requires 3 molecules of CO to produce 1 molecule of G3P, and it involves 3 main phases: carboxylation, reduction, and regeneration of RUBP. Importantly, photorespiration can reduce the efficiency of photosynthesis, especially in sea plants, due to rubisco's oxygenase activity. To minimize this, sea photosynthesis and CAM pathways evolve. Sea plants like maize use spatial separation by fixing CO in mesophyll cells via PEP carboxylase and shuttling malate to bundle sheath cells. CAM plants temporally separate the steps, fixing CO at night and carrying out the Calvin cycle during the day. These adaptations are crucial for photosynthesis under conditions of high light, temperature, or drought. Aerobic metabolism, oxidative harvesting of energy. Aerobic metabolism refers to the complete oxidation of glucose into CO and water in the presence of oxygen, yielding maximum ATP through a tightly regulated sequence of steps. It begins with glycolysis, where one glucose molecule is broken down in a cytosol into 2 pyruvate molecules, generating a net gain of 2 ATP and 2 NADH. The pyruvate is transported into mitochondria and converted into acetyl-CoA, which enters the Krebs cycle, citric acid cycle, in the mitochondrial matrix. In the Krebs cycle, acetyl-CoA is fully oxidized to CO, and high-energy electron carriers NADH and FADH are produced. These reduced coenzymes donate electrons to the electron transport chain, etc., located on the inner mitochondrial membrane. The etc. components, including NADH dehydrogenase complex I, cytochrome BC complex 3, cytochrome C, and cytochrome oxidase complex IV, sequentially transfer electrons to molecular oxygen, the terminal electron acceptor, reducing it to water. This electron transfer drives protons across the inner membrane into the intermembrane space, creating an electrochemical gradient. The proton motive force thus generated powers ATP synthase complex V to phosphorylate ADP into ATP, a process known as oxidative phosphorylation. One glucose molecule under aerobic conditions yields approximately 36 to 38 ATP molecules. This process is tightly regulated through feedback inhibition and allosteric regulation of key enzymes like phosphofructokinase and pyruvate dehydrogenase. Mitochondria play a central role in aerobic metabolism, not only producing ATP but also regulating apoptosis, ROS generation, and metabolic signaling. Evolutionarily, mitochondria are thought to have originated from alpha proteobacteria via endosymbiosis, and the retention of a semi-autonomous genome reflects this ancestry. Mutations in components of oxidative phosphorylation are linked to metabolic disorders and aging. Biological interlink, photosynthesis and aerobic respiration. Photosynthesis and aerobic respiration are intricately connected. The glucose and oxygen produced during photosynthesis serve as substrates for aerobic respiration, while the CO and water generated during respiration are raw materials for photosynthesis. This biogeochemical interdependence supports the global carbon cycle and oxygen balance. In terms of energy transformation, photosynthesis is endergonic, stores energy, while respiration is exergonic, releases energy. In evolutionary terms, photosynthesis likely evolved in terms of energy transformation, photosynthesis is endergonic, stores energy, while respiration is exergonic, releases energy. In evolutionary terms, photosynthesis likely evolved in anoxygenic bacteria before oxygenic photosynthesis arose in cyanobacteria, leading to the Great Oxygenation Event. This oxygen influx allowed aerobic metabolism to evolve, which is far more efficient than anaerobic pathways. These two processes together underscore life's ability to adapt to and reshape the Earth's environment. CSIR-NET Previous Year Questions. CSIR-NET June 2018. In sea plants, the primary fixation of CO takes place in which of the following cells and via which enzyme? A. Bundle sheath cells, via rubisco. B. Mesophyll cells, via PEP carboxylase. C. Guard cells, via carbonic anhydrase. C. Palisade cells, via pyruvate kinase. Answer: B. Mesophyll cells, via PEP carboxylase. CSIR-NET December 2015. During aerobic respiration, the majority of ATP is produced by A. Glycolysis. B. Krebs cycle. C. Substrate level phosphorylation. D. Oxidative phosphorylation. Answer: D. Oxidative phosphorylation. CSIR-NET June 2020. Which of the following statements best describes the functional role of cytochrome C in mitochondria? A. Transfers electrons from complex I to complex IVB, pumps protons into the matrix. C. Transfers electrons between complex 3 and IVD, oxidizes NADH directly. Answer: C. Transfers electrons between complex 3 and IV. Would you like to proceed to the next topic, such as Calvin cycle regulation, photorespiration, oxidative phosphorylation mechanisms, or a new topic from your CSIR-NET Life Sciences syllabus? Just name it and I'll deliver the same high-level notes. You said. Photosynthesis and aerobic metabolism in relation to evolution. ChatGPT said. Here's the CSIR-NET JRF Life Sciences high-level lecture on photosynthesis and aerobic metabolism in relation to evolution. It is crafted as a paragraph only, maximum detail, exam-focused note, building from fundamental to advanced levels. This lecture integrates molecular, cellular, and evolutionary biology, highlighting how photosynthesis and aerobic respiration have evolved and transformed life in Earth's atmosphere. 2 to 3 previous year questions with year and answers are added at the end, as per your request. Photosynthesis and aerobic metabolism in relation to evolution. The evolution of photosynthesis and aerobic metabolism marks one of the most pivotal transitions in the history of life on Earth. These metabolic processes not only represent biochemical innovations but also serve as major evolutionary forces that transform the biosphere. Photosynthesis, particularly oxygenic photosynthesis, fundamentally altered the composition of Earth's atmosphere, enabling the rise of aerobic respiration and multicellular life. The earliest life forms on Earth are believed to have been chemoautotrophs or anaerobic heterotrophs that extracted energy from prebiotic organic molecules or inorganic compounds in the absence of oxygen. These primitive organisms operated in a reducing atmosphere, rich in methane, ammonia, and water vapor. However, as organic molecules became limited, a major selective advantage emerged for organisms that could utilize light energy. This led to the evolution of anoxygenic photosynthesis, likely first developed by green and purple sulfur bacteria. These bacteria used electron donors such as HS instead of water and did not release oxygen. The evolutionary leap to oxygenic photosynthesis, which uses water as the electron donor and produces oxygen as a byproduct, occurred in cyanobacteria. This transformation is believed to have occurred around 2.4 billion years ago, a time associated with the Great Oxygenation Event. The accumulation of atmospheric O due to cyanobacterial photosynthesis had profound evolutionary consequences. First, it led to the oxidation of iron in oceans, forming banded iron formations. Second, it caused a massive extinction of obligate anaerobes, initiating selective pressure for aerobic metabolism. Finally, it laid the foundation for the evolution of complex, energy-demanding eukaryotic cells. The oxygen released from photosynthesis enabled the evolution of aerobic metabolism, a much more efficient energy harvesting process than anaerobic glycolysis. While glycolysis yields a net of 2 ATP per glucose molecule, aerobic respiration yields approximately 36 to 38 ATP. This increase in energetic efficiency allowed the evolution of larger genomes, more complex proteins, intracellular compartmentalization, and multicellularity. Aerobic respiration likely evolved first in ancient prokaryotes that acquired the ability to use oxygen as the terminal electron acceptor in their electron transport chains. The evolutionary origins of mitochondria, the central organelles of aerobic metabolism in eukaryotes, are explained by the endosymbiotic theory. According to this theory, an ancestral anaerobic eukaryotic host cell engulfed an aerobic alpha proteobacterium, which then became a symbiotic organelle, mitochondrion. This symbiosis provided the host with a robust energy source, while the engulfed prokaryote gained protection and access to substrates. Similarly, chloroplasts, the organelles responsible for photosynthesis in plants and algae, are believed to have originated from a cyanobacterium through a secondary endosymbiotic event. The coevolution of photosynthesis and aerobic respiration created a global redox cycle, linking autotrophic production and heterotrophic consumption. Photosynthesis fixes atmospheric CO into organic carbon using solar energy, while aerobic respiration oxidizes this organic matter back to CO, driving the carbon cycle. The rise in oxygen levels also contributed to the formation of the ozone layer, shielding life from harmful UV radiation and enabling colonization of terrestrial environments. On a molecular level, key proteins and genes involved in these processes show signs of conserved evolution. The reaction centers of photosystems e.g., P680 and PSII and P700 and PSI, and cytochrome complexes in the mitochondrial etc. show homology across taxa, indicating a common ancestral origin. Evolutionary tinkering, such as gene duplications, horizontal gene transfers, and endosymbiosis, further contributed to the diversification of these pathways. Additionally, the evolution of C photosynthesis and CAM metabolism in certain plants represents adaptive evolution in response to environmental pressures such as drought, high temperature, and low CO concentration. These adaptations showcase how evolutionary processes refine and optimize core metabolic pathways in different lineages. In summary, the evolutionary emergence of photosynthesis and aerobic respiration fundamentally reshaped life's trajectory on Earth. Photosynthesis supplied oxygen and complex organic molecules, enabling aerobic respiration and the explosion of biological complexity. Together, these two processes illustrate the intimate link between molecular evolution, environmental change, and organismal innovation, forming the energetic and evolutionary backbone of the biosphere. CSIR-NET Previous Year Questions. CSIR-NET Deck 2017. Which of the following statements correctly explains the evolutionary significance of the Great Oxygenation Event? Go. A. It caused mass extinction of cyanobacteria. B. It led to the rise of anaerobic respiration. C. It provided oxygen needed for aerobic metabolism and ozone formation. D. It reduced the energy efficiency of organisms. Answer. C. It provided oxygen needed for aerobic metabolism and ozone formation. CSIR-NET June 2016. According to the endosymbiotic theory, mitochondria are evolutionarily derived from archaea. B. Cyanobacteria. C. Alpha proteobacteria. D. Spirochetes. Answer. C. Alpha proteobacteria. CSIR-NET Deck 2019. Which of the following is a direct evolutionary consequence of oxygenic photosynthesis? A. Decrease in atmospheric CO. B. Increase in anaerobic respiration. C. Formation of the ozone layer. D. Development of RNA world. Answer. C. Formation of the ozone layer. Let me know if you'd like to proceed with another CSIR-NET Life Sciences topic. I'll maintain this same detailed, exam-oriented format throughout.